Human evolution written in our bodies, the fascinating story of vestigial structures. Welcome back to Medical News Pulse, where we explore the incredible intricacies of human biology and the latest developments in medical science. I'm Dr. Scott, and today we're embarking on a remarkable journey through our own anatomy to discover the evolutionary roadmap written within our very bodies. Every structure in the human body tells a story of millions of years of evolutionary adaptation, survival, and change. From the top of our heads to the tips of our toes, we carry within us the remnants of our ancestral past, structures that once served crucial functions for our evolutionary predecessors but have since been modified, reduced, or repurposed as we adapted to new environments and lifestyles. Today, we'll explore these fascinating evolutionary remnants known as vestigial structures, biological features that have lost most or all of their original function through the process of evolution. These anatomical time capsules provide us with extraordinary insights into our evolutionary history and demonstrate how our bodies have adapted over millions of years to create the remarkable biological machine that is the modern human form. Understanding vestigial structures, evolutionary footprint. The term vestigial derives from the Latin word vestigium, meaning footprint or trace. These structures represent the biological equivalent of archaeological artifacts, remnants of our evolutionary past that persist in our modern anatomy despite having lost their original purpose. Understanding these features provides us with a unique window into the evolutionary processes that shaped our species. Vestigial structures differ significantly from rudimentary organs, which are underdeveloped versions of functional structures found in related species. True vestigial features have undergone a process called evolutionary reduction, where natural selection has gradually diminished their size and function over countless generations because they no longer provide survival advantages in current environments. The study of vestigial anatomy has revolutionized our understanding of human evolution and our relationship to other species. These structures provide compelling evidence for common ancestry and demonstrate how evolutionary pressures shape organisms over time. Rather than being perfectly designed from scratch, our bodies represent the result of countless modifications and adaptations built upon pre-existing structures inherited from our ancestors. The auricular muscles. When ears could move. Beginning our anatomical journey at the head, we encounter one of the most intriguing examples of vestigial musculature, the auricular muscles surrounding our ears. These small muscle groups, consisting of the anterior, superior, and posterior auricular muscles, represent remnants of a once powerful system that allowed our ancestors to move their ears independently to better locate sounds in their environment. In most mammals, including our closest relatives among the primates, ear movement serves as a crucial survival mechanism. Dogs, cats, horses, and many other animals can rotate their ears nearly 180 degrees, allowing them to pinpoint the location of sounds with remarkable precision without moving their heads. This ability provided significant advantages for detecting predators, locating prey, or communicating with other members of their species. Modern humans retain these muscles, though they have become significantly reduced in size and functionality. Only a small percentage of the population can demonstrate any voluntary ear movement, and even then, the range of motion is extremely limited compared to our evolutionary relatives. However, fascinating research has revealed that these muscles remain neurologically connected to our auditory processing centers. Neurological studies using brain imaging technology have demonstrated that when humans hear sounds, particularly those coming from specific directions, the brain areas controlling the auricular muscles show measurable activity. This suggests that although we can no longer move our ears effectively, our nervous system still attempts to orient them towards sounds, a ghostly echo of an ancient survival mechanism that once helped our ancestors navigate dangerous prehistoric environments. The Plica-semilunaris, remnant of a third eyelid moving to the eyes, we discover another remarkable vestigial structure that reveals our connection to the broader animal kingdom. Located in the inner corner of each eye, the Plica semilunaris appears as a small, crescent-shaped fold of tissue that most people notice but rarely consider. This seemingly insignificant structure represents the evolutionary remnant of what biologists call the nictitating membrane, a third eyelid that moves horizontally across the eye to provide additional protection and moisture. This transparent or translucent membrane remains fully functional in numerous animal species, including birds, reptiles, sharks, and many mammals. The nictitating membrane serves multiple crucial functions in animals that retain it. It provides protection from debris, sand, and wind while maintaining vision, offers additional moisture to the eye surface, and can even function as a natural goggle for aquatic animals. Birds of prey use their nictitating membranes to protect their eyes while diving at high speeds, while sharks employ them during feeding to prevent injury from struggling prey. Interestingly, this structure appears to have been lost independently in both humans and our closest primate relatives, the chimpanzees, suggesting that our common ancestor also lacked a functional third eyelid. This evolutionary loss likely occurred because our ancestors developed sufficient eye protection and moisture production through other means, including improved hair production and behavioral adaptations. Wisdom teeth, remnants of ancient dietary needs. Perhaps no vestigial structure causes more contemporary problems than our third molars, commonly known as wisdom teeth. These four teeth, located at the very back of the dental arch, represent one of the most problematic examples of evolutionary mismatch between our ancestral heritage and modern anatomy. To understand why wisdom teeth have become such a source of dental difficulties, we must examine the dramatic changes in human jaw structure and dietary habits that occurred during our evolutionary history. Our early ancestors possessed much larger jaws and stronger chewing muscles, adaptations necessary for processing the tough, fibrous plant materials that comprised a significant portion of their diet. The powerful molars served as grinding surfaces for breaking down leaves, tough roots, bark, and other plant materials that required extensive mechanical processing before swallowing. Fossil evidence from early hominids reveals dental wear patterns consistent with a diet requiring substantial chewing force and time, very different from the processed foods that characterize modern human nutrition. As human evolution progressed, several factors contributed to the reduction in jaw size and the problematic nature of wisdom teeth. The development of tool use allowed our ancestors to process food externally, reducing the need for powerful chewing apparatus. The controlled use of fire for cooking further softened foods and reduced the mechanical demands on our dental system. Simultaneously, dietary changes toward more omnivorous eating patterns, including increased consumption of animal proteins, reduced the need for extensive plant processing. The evolutionary flattening of the human face, which gave rise to our distinctive facial structure and prominent chin, compressed the available space for teeth. This architectural change, while providing advantages in other areas such as speech production and social communication, created insufficient room for the full complement of ancestral teeth. The result is that wisdom teeth frequently become impacted, growing at abnormal angles or failing to emerge properly, necessitating surgical removal in many individuals. Male nipples, developmental remnants. One of the most commonly questioned vestigial structures in human anatomy is the presence of nipples in males. This feature provides an excellent example of how developmental biology and evolutionary history intersect to create structures that may seem purposeless but actually result from fundamental aspects of mammalian development. The presence of nipples in males results from the basic mammalian developmental template that establishes body structure before sexual differentiation occurs. During early embryonic development, all mammals follow essentially the same developmental pathway for the first several weeks of gestation. Nipples form during this early period, before the hormonal changes that determine male or female characteristics begin to influence development. From an evolutionary perspective, male nipples represent what biologists call a developmental constraint, a structure that exists not because it serves a current function, but because it develops as part of a shared mammalian body plan. The genetic and developmental machinery required to eliminate nipples in males would likely interfere with their proper formation in females, so natural selection has maintained the current developmental pattern. Interestingly, male nipples retain their neural connections and can respond to stimulation, demonstrating that vestigial structures need not be completely non-functional. In rare cases, hormonal abnormalities can even cause male breast development and milk production, revealing the latent potential that remains within these seemingly purposeless structures. The evolutionary history of nipple development also provides insights into mammalian ancestry. Many mammals possess multiple pairs of nipples arranged along what developmental biologists call the mammary ridge, or milk line. Occasionally, humans are born with additional nipples along this ancestral pattern, a condition called supernumerary nipples or polythelia, providing direct evidence of our mammalian heritage. Body hair patterns, traces of our furry past. The distribution of hair on the human body provides compelling evidence of our evolutionary transition from a fully furred ancestor to our current relatively hairless state. While humans are often described as naked apes, we actually possess roughly the same number of hair follicles as other primates. The difference lies in the thickness and prominence of the hair produced by these follicles. The evolutionary loss of dense body hair represents one of the most significant adaptations in human evolution, likely related to our ancestors' transition from forest-dwelling creatures to savanna-living, highly active hunters and gatherers. Dense fur, while excellent for temperature regulation in stable environments, becomes problematic for animals that engage in sustained physical activity, particularly in warm climates. The reduction in body hair coincided with significant enhancements to the human sweating system. Humans possess an unusually high density of sweat glands compared to other mammals, and our sweating capacity far exceeds that of our closest primate relatives. This adaptation allowed our ancestors to engage in persistence hunting, a hunting strategy that involved pursuing prey over long distances until the animal collapsed from exhaustion. However, hair has been selectively retained in specific areas of the human body where it continues to serve important functions. Hair on the scalp provides protection for the brain, one of our most temperature-sensitive organs. The brain requires careful temperature regulation to function optimally, and scalp hair helps insulate this vital organ while also protecting it from solar radiation. Hair in the underarm and genital regions serves multiple functions related to scent communication and friction reduction. These areas contain specialized scent glands that produce chemical signals important for social and reproductive communication. The hair in these regions helps trap and disperse these chemical messages while also reducing friction during movement and intimate contact. Goosebumps, the remnant defense mechanism. The phenomenon of goosebumps provides one of the most immediately observable examples of vestigial function in the human body. When we experience cold, fear, emotional arousal, or certain types of aesthetic experiences, tiny muscles called erector pili contract around each hair follicle, causing the hair to stand upright and creating the characteristic bumpy texture on the skin surface. In our evolutionary ancestors and in modern mammals with dense fur, this response served crucial survival functions. When the hair stands erect, it creates an insulating layer of trapped air close to the skin surface, significantly improving the body's ability to retain heat in cold conditions. This mechanism allows mammals to dynamically adjust their insulation based on environmental conditions without requiring behavioral changes. The erector pili response also served as an important display mechanism for communication and intimidation. When a mammal feels threatened, the erection of hair makes the animal appear larger and more formidable to potential predators or rivals. This defensive display can often prevent actual physical confrontations, providing a survival advantage through psychological intimidation rather than physical combat. In modern humans, this response has become largely vestigial because our sparse body hair cannot provide meaningful insulation or create an impressive intimidating display. However, the neural pathways and muscular mechanisms remain intact, demonstrating how evolutionary remnants can persist long after their original function has been lost. Recent research has revealed that the goosebump response may retain some functionality in modern humans through its connection to emotional and aesthetic experiences. The same neural pathways that once triggered this response for survival purposes now activate during intense emotional moments, suggesting that this ancient mechanism has been co-opted for social and cultural communication. The appendix, from useless to essential. The vermiform appendix represents one of the most dramatic reversals in our understanding of vestigial structures. For over a century, this small, worm-like extension of the large intestine was considered the classic example of a useless evolutionary remnant, a structure that served no purpose other than to occasionally become inflamed and require surgical removal. This view of the appendix as completely vestigial has been revolutionized by modern research into the human microbiome and the complex ecosystem of beneficial bacteria that inhabits our digestive system. Scientists now understand that the appendix serves as a crucial reservoir for beneficial gut bacteria, functioning essentially as a safe house where helpful microorganisms can survive and multiply. The appendix's location and structure make it ideally suited for this function. Its narrow opening prevents the rapid flow of digestive contents through the organ, creating a relatively stable environment where beneficial bacteria can establish communities without being constantly flushed away. The appendix is also rich in immune tissue, which helps regulate the bacterial populations and prevent harmful organisms from establishing themselves. During episodes of severe diarrhea, food poisoning, or antibiotic treatment that can devastate the gut microbiome, the bacteria stored in the appendix can rapidly recolonize the intestines, helping to restore normal digestive function and immune protection. This function appears to be particularly important in environments where exposure to waterborne pathogens is common. The evolutionary perspective on the appendix has also shifted dramatically. Rather than being a useless remnant, the appendix now appears to represent an adaptation that became particularly important as our ancestors developed larger social groups and increased exposure to infectious diseases. The ability to rapidly restore gut bacteria after illness episodes would have provided significant survival advantages in these challenging environments. The coccyx, our ancestral tail at the base of the human spine, lies one of our most obvious vestigial structures, the coccyx or tailbone. This small triangular bone, composed of 3 to 5 fused vertebrae, represents the remnant of the tail that our distant ancestors possessed millions of years ago. The evolutionary loss of the tail marks one of the most significant changes that occurred in the lineage leading to modern apes and humans. While many of our primate relatives retain fully functional tails that serve important roles in balance and locomotion, the great apes, including humans, chimpanzees, gorillas, and orangutans, have all lost their external tails. The reduction and eventual loss of the tail in the ape lineage correlates with significant changes in locomotion patterns and lifestyle. Early apes developed a form of movement called brachiation, swinging from branch to branch using their arms, which required different balance mechanisms than the quadrupedal movement typical of tailed primates. As apes grew larger and spent more time on the ground, tails became less beneficial for balance and potentially problematic for terrestrial movement. The human coccyx serves some important functions despite its vestigial status. It provides attachment points for several muscles involved in pelvic floor support, including muscles crucial for bowel and bladder control. The coccyx also helps support body weight when sitting and provides structural integrity to the lower spine. Occasionally, developmental anomalies result in humans being born with external tail-like structures, a condition known as human tails or vestigial tails. These rare occurrences provide direct evidence of our tailed ancestry and demonstrate how evolutionary developmental programs can sometimes revert to ancestral patterns. Vestigial muscles. The changing demands of locomotion throughout the human body, numerous muscles have become reduced in size and function as our locomotion patterns evolved from quadrupedal to bipedal movement. These vestigial muscles provide insights into the dramatic anatomical changes required for upright walking and running. The pyramidalis muscle, located in the lower abdomen, represents one such vestigial structure. This small, triangular muscle appears to serve little function in modern humans and is even absent in some individuals. In other primates, this muscle group plays a more significant role in abdominal support and movement, but the demands of bipedal locomotion have reduced its importance in humans. The plantaris muscle in the lower leg provides another example of evolutionary muscle reduction. This muscle, which extends from the knee to the heel, has become essentially vestigial in humans, existing primarily as a long tendon with minimal muscle tissue. In other primates, the plantaris plays a more significant role in foot flexion and climbing movements, but our specialized adaptations for ground-based locomotion have rendered it largely unnecessary. These muscular changes reflect the comprehensive anatomical reorganization required for efficient bipedal locomotion. Every aspect of human anatomy, from the shape of the pelvis to the structure of the foot, has been modified to support upright walking and running, often at the expense of other capabilities that our ancestors possessed. The human foot, engineering for bipedalism. The human foot represents one of the most dramatic evolutionary modifications in our anatomy, specifically engineered for the unique demands of bipedal locomotion. The development of our characteristic double arch system distinguishes human feet from those of all other primates and provides crucial insights into the evolutionary pressures that shaped our species. The longitudinal arch, running from heel to toe, and the transverse arch, spanning the width of the foot, work together to create a structure that functions like a complex spring system. This arrangement allows efficient energy storage and release during walking and running while providing the stability necessary for upright balance. The evolutionary development of these arches required extensive modifications to bone structure, ligament arrangements, and muscle function. Our closest primate relatives possess relatively flat feet that are well suited for grasping branches and navigating through trees but are inefficient for sustained ground-based locomotion. The condition known as flat feet or pes planus represents a developmental variation where the arch structure fails to develop properly or collapses over time. Individuals with flat feet must use additional muscle energy to provide the stability normally supplied by the arch structure, demonstrating the functional importance of this evolutionary adaptation. Modern footwear, particularly high-heeled shoes, can interfere with the natural function of the foot's arch system. These artificial modifications to foot position can stress the evolutionary adaptations that took millions of years to develop, leading to various foot problems and demonstrating the importance of understanding our anatomical heritage when making lifestyle choices. Conclusion: the living record of evolution, a journey through the vestigial structures of the human body, reveals the remarkable story of our evolutionary heritage. Every seemingly purposeless structure, every reduced muscle, and every architectural compromise in our anatomy tells part of the larger narrative of how we became the unique species we are today. These evolutionary remnants remind us that our bodies are not perfect machines designed from scratch, but rather the result of countless modifications and adaptations built upon the foundation of our ancestral heritage. Understanding these structures provides valuable insights not only into our evolutionary history but also into potential medical issues and the importance of considering our biological heritage when making health decisions. The study of vestigial anatomy continues to evolve as new research reveals previously unknown functions for structures once considered useless. The appendix's role in maintaining gut health, the continued neural connections of ear muscles, and the biomechanical importance of various reduced muscle groups all demonstrate that evolution rarely produces truly purposeless structures. As we advance in our understanding of human biology and continue to uncover the intricate connections between structure and function, these evolutionary remnants serve as constant reminders of our deep connection to the natural world and our place in the grand tapestry of life on Earth. Thank you for joining us on Medical News Pulse for this fascinating exploration of our evolutionary anatomy. Don't forget to subscribe for more insights into the incredible science of human biology, and let us know in the comments which vestigial structure you found most interesting in your own body. If you found this interesting, please like and subscribe to the channel.